Welcome everyone to Research Grand Rounds for UofL Department of Medicine. I'm Dr. Jen Cook, one of our vice chairs, so I'm pleased to introduce our speaker today, Dr. Andrew Gibb. So Dr. Gibb earned his Ph.D. in Physiology and Biophysics here at UofL in 2017, and then he went on an adventure from 2018 to 2023 to Temple University, where he did his postdoc and joined their faculty as an instructor, but we were lucky enough to successfully recruit him back in 2024 to UofL, where he now serves as assistant professor at the Department of Medicine's Division of Environmental Medicine and the Center for Cardiometabolic Science. So in addition to serving as a co-I on an R01, he currently holds an American Heart Association Career Development Award for his work in glutamine metabolism, which is apropos to our subject matter today. And though he's only been with us as an assistant professor for a year, he is already developing a really impressive footprint as a scholar, with not only through his publications, but also through a really impressive number of international presentations. So I'd like to welcome Dr. Gibb to Department of Medicine Research Grand Rounds, and we look forward to learning about glutamine metabolism with you today.
Thanks.
Mm-hmm.
Thank you for the introduction, and thank you to the Department of Medicine and Dr. Williams for allowing me to present here today. So today I'm going to talk to you about some of the previous presented work and published work that was attained during my postdoc time, and then some of the new projects that I'm currently developing now that are leading to the establishment of my lab. So there are a lot of cell types that make up the heart. Two of these that are most important and what I'm going to be talking about today are the cardiomyocytes themselves and the fibroblasts. And it's not surprising that these are really important to cardiac physiology and pathophysiology, as they make up about 65% of the total cells within a heart. So in the first project that I'm going to the first half, I'm going to be talking about the role of glutamine metabolism in the fibroblast compartment. And then the second half of the talk, we'll be looking at glutamine metabolism and the cardiomyocytes themselves, and then hopefully trying to link these two together in some type of potential crosstalk mechanism. So cardiac fibrosis is a very central key feature of both heart failure with reduced ejection fraction and preserved ejection fraction. There are multiple different types of fibrosis that occur, but overall, this is an excessive deposition of extracellular matrix that contributes to overall deleterious modeling. And so this really ends up reducing compliance, impairing conduction, disrupting cell-to-cell communication, ultimately leading to the development of heart failure. As you can see here, this is a mouse model of heart failure. In our sham-operated mice, we see no red staining, which is indicative of collagen and fibrosis. But when we subject these mice to a pressure overload model, we can see a significant infiltration of this collagen deposition. And there's been a lot of causal studies that have suggested the vast majority of the extracellular matrix is derived from this fibroblast population that is resident to the heart. And as I mentioned, this really impacts function. As you can see here in our sham-operated mice, these hearts are able to contract quite well. And you can see that this ability to contract and pump blood is significantly impaired in the pressure overloaded animal. So as I mentioned, we believe that, or it's pretty well defined, that the fibrotic response is originated from endogenous cardiac fibroblasts that are located in the heart. And you can see that by this lineage tracing study, where they used a lineage marker of TCF21, which labels cardiac fibroblasts. And you can see the degree of expansion that this occurs in response to pressure overload. Similarly, these TCF21 cells ultimately, upon activation, start to express this protein periostin de novo. And using additional periostin lineage tracing experiments following MI, this entire fibrotic response is due to this increase in periostin in this cell type. So what are these fibroblasts? Well, they're really important just structurally and homeostatically in the naive heart. They contribute to just general ECM maintenance and turnover and electrical conduction. But when they become activated upon stressors such as pressure overload or profibrotic agonists such as transforming growth factor beta or angiotensin 2, they become activated. And this results in this proliferative burst, an increase in different adhesion complexes, extracellular matrix proteins, and as well as the production and secretion of this periostin protein, as I was mentioning. So this is really a signature of activated fibroblasts, which will be important as I move through this talk. Now, as we know, heart failure and a lot of these stressors tend to be chronic. So upon sustained stress, these fibroblasts move out of this activated state into this differentiated state, increasing alpha smooth muscle actin, the stress fiber network, increasing extracellular matrix proteins even more, and they're really resistant to cell death. So this really contributes to their persistence in the heart. And there's been some literature, which I'll show as well, with some of my results, that we may actually be able to target this cell population to de-differentiate them and get them back to a more quiescent and unactivated state. So when I joined my postdoc lab, I was involved in a few studies that were really focusing on the pathways and the processes that activate fibroblasts and trying to intervene to see if that is a therapeutic target. What we were able to show is that if we stimulate cells with profibrotic agonists, they increase the expression of this protein called MICU1, which I'm not going to get too much into, but basically it reduces mitochondrial calcium uptake. And what that results in is an increase in alpha-ketoglutarate, which is a metabolite within the TCA cycle. And this led to transcriptional, epigenetic and transcriptional changes within the nucleus to activate gene transcription. And so if we intervened there and prevented this from happening, then we would prevent the fibroblasts from differentiating. However, there's a couple questions that kind of came from this. We saw changes in metabolism with this TGF-beta response, but we really weren't sure what substrates or what fuels were contributing to the myofibroblast formation, or where these substrates were coming from, and if we could target these metabolic pathways specifically. And so to try to get at this, I treated cells, cardiac fibroblasts, with TGF-beta using a carbon tracer for either glucose or glutamine. And so what this allows us to do is kind of track where these carbons go into certain metabolites so that we can really understand how pathways change. And so as you can see here, really simply, if we just look at this 0 time point, a decrease indicates that the cells that are treated with TGF-beta in the red, there's more activity and there's more synthesis of that metabolite. And so as you can see, this indicates that glycolysis is overall increased when we stimulate cardiac fibroblasts with TGF-beta. Interestingly, trying to understand that role of alpha-ketoglutarate, we didn't really see any carbons from glucose ending up in the Krebs cycle or in alpha-ketoglutarate, suggesting that glucose oxidation doesn't really occur in these cells. So from additional experiments that we had done, we looked at the role of potentially glutamine as a metabolite that could be serving the biosynthesis of alpha-ketoglutarate, which would lead to all of those transcriptional changes to drive the program. And so as you can see here, when we labeled with glucose, we did this for 12 hours. But when we only label with glutamine for 1 hour, we can see a significant increase in the amount of alpha-ketoglutarate that's generated from glutamine in response to TGF-beta. And so this really indicates that glutamine is a significant substrate for alpha-ketoglutarate biosynthesis and for oxidative metabolism. And then we also measured this at the absolute level, and we can see, more than a 2-fold increase in alpha-ketoglutarate abundance in the cardiac fibroblasts. So in addition to glutamine contributing to alpha-ketoglutarate biosynthesis, it's likely that it can also contribute to extracellular matrix and fibroblast function in additional ways, such as collagen biosynthesis, proline biosynthesis. These are really important processes for remodeling the extracellular matrix, as well as like glutathione processes has been shown to regulate the myofibroblast phenotype as well. And so we just asked a very simple question, seeming that glutamine seems to be really important, is the breakdown of glutamine necessary for myofibroblast formation, collagen synthesis, and the eventual fibrotic response in the heart? And so we can see here that in response to TGF-beta, we don't really see any change in the glutamine, the glutaminase enzyme, the enzyme that breaks down glutamine to glutamate. And we didn't really see any change transcriptionally either. So we think that this increase in glutamine synthesis is just a mass action event where it just is consuming, but not necessarily changing the machinery. So to start to intervene and test whether the breakdown of glutamine, which I'm terming glutaminolysis, is necessary for myofibroblast formation, we used a pharmacological inhibitor, CB839, which targets that first step, the glutaminase gene. And this is a really important and powerful pharmacological inhibitor because this is currently being used in phase 2 clinical trials for cancer research. So it could potentially be, if it was found to work out well, could potentially be a means that we could repurpose this for fibrosis. So if we use this inhibitor, we can see if we compare our black to our blue bars, which is just the inhibitor alone, we can see that we get an accumulation of glutamine in the fibroblasts, and we see a reduction of the downstream metabolites, glutamate and alpha-ketoglutarate, indicating that it's working. If we look at our TGF-beta treated cells, we can see that there's an increase in glutamate, an increase in alpha-ketoglutarate. As we would expect, this process is increased, but when we use our inhibitor, we're able to blunt this effect. So looking at different processes and different assays that we can test for myofibroblast differentiation, you can see here with TGF-beta, we get this increase in collagen, an isoform of collagen, which is really important in the extracellular matrix. We can see the increase in the expression of alpha smooth muscle actin, which I said was a de novo marker of a myofibroblast. And these cells are able to contract kind of these gel matrix plugs that we artificially embed the cells in. And then if we use our pharmacological inhibitor in the presence of TGF-beta, you can see that we blunt that increase in collagen, we blunt that increase in alpha smooth muscle actin, and we prevent that ability for these cells to contract a gel matrix, suggesting that inhibiting glutamine metabolism is sufficient to prevent these cells from differentiating. We also looked at metabolism. We see that in response to TGF-beta, we get this large increase in oxidative metabolism using a seahorse extracellular flux analyzer. So this just measures oxygen consumption by the cells. And you can see if we compare our green to our red here, using the inhibitor in the presence of TGF-beta completely prevents that response. So because pharmacological targets aren't always specific, we also wanted to look if this was a specific event to the GLS1 gene. And so we used a genetic approach to delete the gene within the cells and confirm that here by protein expression that we no longer have the glutaminase gene. As shown before, we get this nice increase in alpha smooth muscle actin expression with TGF-beta. This nice ability to contract these collagen gel matrix plugs that we embed the cells in. And if we delete the gene, we prevent that response from occurring. So as we previously showed as well, we showed that the alpha-ketoglutarate is this substrate that is used for histone demethylation events. And typically histone demethylation is accompanied by acetylation, and that really is a permissive and active mark for gene transcription. And so part of our hypothesis was that by preventing the accumulation of alpha-ketoglutarate, then we prevent this demethylation and then the eventual prevention of the fibrotic gene program activation. And so as you can see here with TGF-beta, we see this decrease in histone methylation status. And we, again, blunt that with our inhibitor in the presence of TGF-beta. So next, we did a rescue experiment where we treated cells with dimethyl alpha-ketoglutarate. So alpha-ketoglutarate it's amino acid, it's charged, it can't be taken up by the cell, there's no transporters for it. So by adding this dimethylation on it, this allows it to become cell permeable and the cells can take it up. And so if we use our pharmacological inhibitor and rescue back the ability of the cells to have an excess of alpha-ketoglutarate, the cells are able to differentiate and become a myofibroblast. So this is all great. We can find this in mice and it works, very nice and perfectly, but could this actually be something that could occur in human heart failure? So when I was at Temple University, I was fortunate enough to collaborate with Dr. Ken Margulies, who's a clinician physician at UPenn. And we were able to isolate cardiac fibroblasts from left ventricle biopsies of non-ischemic heart failure patients. And so they were obtaining these. We would enzymatically dissociate and isolate and plate these cells, treat them with TGF-beta, use our pharmacological inhibitor, and then study whether these cells became differentiated. And as you can see here, across 3 different heart failure explant cardiac fibroblasts, you can see that in all of these patients, we were able to, or all of these samples, we were able to prevent that ability of the cells to express alpha smooth muscle actin de novo in response to TGF-beta. And when we look at gene panels of common fibrotic gene sets, we can again see that this nice increase with TGF-beta, black versus red. But then for a lot of these ones where we use our GLS-1 inhibitor, we can prevent this response as shown by the bars in green. So again, as I mentioned, that's really important. This seems to be something that could potentially be translational, but we're not going to treat patients or treat any of this before disease sets in. So the question then becomes, can we actually reverse the myofibroblast phenotype? And if so, can in vivo targeting of this specific pathway of glutamine metabolism, could this decrease fibrosis and heart failure progression? And so here we did another in vitro experiment where we treated cardiac fibroblasts with TGF-beta for 48 hours, allowed them to get to a point where they are expressing alpha smooth muscle actin and becoming differentiated. And then we intervened with our pharmacological inhibitors or by removing glutamine from the media. And you can see here that with TGF-beta, for that 96 hours, the cells are differentiated. They express the smooth muscle actin. But when we remove or when we intervene at the 48-hour time point, those cells become inactivated. So to try to start approaching this in an in vivo model, we used a genetic approach in which using our GLS-1 conditional knockout mice, we crossed those with a periostin inducible Cre, and then again with a Cre-dependent TD tomato reporter. And so what this allows us to do is to fluorescently label any activated fibroblasts only when they're activated. And then also at the same time, simultaneously delete the gene for GLS-1 only again in those activated myofibroblasts. So we did that. We subjected these mice to TAC. So they're on this tamoxifen chow, which allows for this whole genetic process to undergo. And then serial echocardiographies were obtained to assess cardiac function. And as during this protocol, as the fibroblasts become differentiated and activated, they will become red and they will also become deficient of GLS-1. And so as you can see here in a small cohort that we took at the end of the study, we fluorescently sorted the cell population for the red cells and then did a Western blot for GLS-1. And you can see that there's no longer expression in this specific cell population as compared to the controls. It's important that we made sure that the stimulus between our 2 groups was the same. So what we did here was at the time point that we induced TAC, we ensured that we had an increase in the peak pressure gradient across the aorta. And you can see that between our 2 genotypes, there's no difference, suggesting that the insult and the stress that they were fighting against was equal. Now, initially, we didn't see really any difference between our 2 genotypes. Knockout of GLS-1 didn't really seem to make a difference up to about 8 weeks. But then after that, you can see that the groups start to separate. And the ones that the cohort or the group that had the deletion of GLS-1 exclusively only in the activated and differentiated fibroblasts seemed to do better and have a better cardiac function. We also looked at the strain using B-mode speckle tracking with our echocardiography. And as you can see here, strain or ventricular deformation was improved and enhanced in this population that had the GLS-1 deleted. as well as the inverse strain, which this is really important because this is kind of more correlated with diastole and relaxation, and that aspect of cardiac function and ventricular deformation is really tightly associated with tissue fibrosis. So to measure tissue fibrosis directly, we used picoseries red staining, which again, as I showed in I think the first slide, stains anything, any collagen red. So you can see with our pressure overload, our TAC animals, we have this significant increase in collagen, and in our deleted GLS1 deleted animals, again, exclusively only in the myofibroblasts, we see that as prevented. And then to try to understand, why we kind of saw this effect where this only kind of occurred late and the group started to separate, we figured that this was some kind of time-dependent thing, where, there's a time limit and a time component of getting this protein to turn over and actually be deleted from those cells. And so at an earlier time point, 6 weeks, we can actually see that there's a pretty similar degree of fibrosis between our two genotypes. But then at the 16-week time point, you can see there's a significant reduction in the tissue fibrosis only in those animals where we deleted GLS1. And so this really not only shows that, we're getting a reduction in the fibrotic response, but more so that this is actually a reversal of the fibrotic response, which provides the most clinical and relevant physiological importance. So again, to see if this reversal phenotype is something that we could reproduce in the human failing heart, we again took our patient LV biopsy cardiac fibroblasts and again treated these with TGF-beta for 24 hours, then intervening, again doing that kind of allowing the cells to differentiate, then intervening and seeing if we can decrease the expression of these myofibroblast markers. And in these two patient samples here, you can see that, again, while we have this activation in the TGF-beta treated cells, this was reversed with our inhibitor and as well at the gene level. So taken together, it seems that if we target glutaminolysis in these myofibroblasts, that this is sufficient to reverse their phenotype and could provide a new target for treating active fibrosis and heart failure progression. So to kind of switch gears just a little bit, I'm going to switch over to the potential role of glutamine in the cardiomyocytes themselves. And so we've had a lot of research endeavors to increase energy supply in heart failure. And I don't think I need to tell many that part of the issue with the failing heart is that it becomes energy starved and it starts to lose the ability to contract and function properly. And part of this is tied to poor energetics. And we've done a lot of things to try and affect fatty acid metabolism, glucose metabolism. Most recently, there's a lot of research that's focusing on ketone metabolism or branched chain amino acids. But very little research, if any, has been done on the role of glutamine or glutamate metabolism in the context of cardiac metabolism. And this is really kind of strange to me because glutamine is the most abundant amino acid in circulation. And so it seems like it would be a very easy substrate for the heart to use. I don't know. What happened? Oh.
I don't know. Hopefully we just fixed it.
Okay, we're back.
Okay.
All right. I think it's sharing still, so I think we're okay. Yeah. All right, thanks. So this leaves a very novel and interesting area to understand how glutamine metabolism, what impact that has in the cardiomyocytes in the context of heart failure. So there's a lot of potential roles, transcription, translation, bioenergetics, and NAD biosynthesis, which is intricately linked to bioenergetics, the redox potential. And so those are kind of some of the ones that currently right now I've got preliminary data that would suggest these have an important role within the context of heart failure. So as I mentioned, I talked a lot about glutaminase, which is the enzyme that breaks down glutamine to glutamate. But there's another enzyme called glutamine synthetase, which actually adds back that ammonia to glutamate to form glutamine. Now, interestingly enough, glutamine synthetase is expressed primarily in highly energetic tissues. Specifically, the skeletal muscle and the heart actually express some of the most abundant tissues of glutaminase. And so, interesting though, we would expect that the heart would just use what it can, but it actually can't. So, work by Heinrich Tagmeyer actually showed that there was very little glutamine that was converted to glutamate, but that the heart actually converted a lot of glutamate to glutamine. And in a more recent paper here by Dr. A.J. Shaw, he showed that in response to pressure overload, that same TAC model that we used, that more glucose labeled glutamate and glutamine, indicating that glutamine synthesis is increased in the failing heart. And more importantly, in a more recent study from Dr. Zoltarani's lab, one of our collaborators at UPenn, what they did here was they looked at metabolite differences between the coronary sinus and the radial artery to look at what was going in and what was coming out of the heart. And of all the metabolites that was released from the heart, glutamine was the highest, suggesting that the heart actually synthesizes and secretes glutamine, and that this appears to be increased in the failing heart. When we look at some of our data sets to see whether glutamine synthesis its expression is affected in heart failure, we see that in our pressure overload model, the expression of glutamine synthetase is increased. That's GLUL here, as well as in this rat model of hypertension where we see an increase in expression. And so there's a couple of means by which glutamine synthetase could impact cellular bioenergetics. Specifically, one hypothesis is that the net flux of synthesis limits the ability of the cardiomyocytes to actually use glutamine or glutamate for ATP provision. Another possibility that we have some preliminary data on is that glutamine synthetase activity could and likely does modify fuel utilization. So the activity of this enzyme could influence how the cell uses fatty acids or glucose or other substrates. And then another leading hypothesis that we're working on right now is that in heart failure, an increase in glutamine synthetase activity could actually act as like an energetic siphon, pulling carbons from the Krebs cycle that ultimately we want to use for energetics. But because this process is increased, it actually becomes a maladaptive effect. And so the idea is by targeting glutamine synthetase in the cardiomyocytes, could this provide a therapeutic benefit, and would that be through increasing energy metabolism? And so we created a novel conditional glutamine synthetase knockout by crossing this with the cardiomyocyte-specific alpha myosin heavy chain Cre. And as you can see here by this Western blot, we've confirmed that we've effectively deleted this gene at the protein level. Also, to confirm that this is working, we measured the abundance of glutamate and glutamine and the conversion of these two metabolites. And you can see here that with deletion of the gene, glutamate levels rise within the heart. Glutamine levels fall a little, but non-significantly, probably just because they're still taking it up from the circulation because it's so abundant. But when we use a carbon tracing approach, we can see that there are less carbons that go from glutamate to glutamine, indicating that we've effectively reduced this pathway. When we look at oxidative and glycolytic metabolism in isolated adult cardiomyocytes using this extracellular flux analyzer, we can see that basally the knockout cells, knockout cardiomyocytes, are more energetically capable. They have a higher respiratory rate as well as a higher glycolytic rate. And when we inject the uncoupler called FCCP, this basically drives the electron transport chain to work as fast and as hard as it can. And we can see then the knockouts, they have a larger response, a greater response, indicating that they can likely meet a higher energetic demand when needed. Similarly, when we inject oligomycin, this significantly increases glycolysis by inhibiting ATP production within the mitochondria. And we can see that the knockout cardiomyocytes, again, are able to activate a greater response of glycolysis in this to this response. To try to tease out whether this basal difference and whether these responses to these uncouplers or activators of glycolysis is substrate driven. We again took our two genotypes in the adult cardiomyocytes and substrate deprived them for 1 hour prior to running this assay. And you can see here that there's no basal difference now within oxidative metabolism or glycolysis, indicating that this difference was truly substrate driven. And if we inject just glucose and see how these cells respond only to glucose, we can see an increased rate of oxidative metabolism and an increased rate of glycolysis. So they're more capable and able to use glucose as an energy substrate. Again, in vivo, to show that this is occurring in vivo, we used stable isotope tracing approaches and see that there's more glucose that ends up in lactate, indicating that glycolysis is truly elevated in the knockout heart. We also looked to see if loss of GS, try to pair this with the seahorse data, if this increased the energetic state of the heart. We didn't really see any change, although some of these metabolites trended to be higher in the knockouts with the Krebs cycle. But what we did find, surprisingly, was that all these high energy intermediates, ADP, ATP, the ATP to ADP ratios, nicotinamide, and creatine were all significantly higher in the knockouts, suggesting that the knockout heart is just at a better energetic state than the wild type. And so collectively, as I mentioned, all these measurements suggest that the GS, loss of GS increases cardiomyocyte bioenergetics and that they're at this higher cardiac energy state, which we would hypothesize would be very beneficial in the failing heart. And so to test whether this is beneficial in the failing heart, which is energy starved, we used our knockout mice crossed on the Cre-dependent reporter for the cardiomyocytes and then appropriate controls, and subjected them to the pressure overload model for 12 weeks with serial echoes every 4 weeks. Again, we assessed to make sure that deletion of GS had no baseline effect in sham mice. 12 weeks post deleting the gene, there's no change in cardiac function, so they seem to be fine. However, in response to the pressure overload, you can see here, represented by fractional shortening, that the knockouts do significantly better in response to this pressure overload stimulus, where the Cre controls just continuously develop heart failure and worsening function. And we think that it appears that this is more due to a improvement in systolic function, which again would be very easily explainable by an increased energy state. Again, just to kind of further characterize what we were able to see, we saw that cardiomyocyte-specific deletion had no real impact on the hypertrophic response in DETECT, but we did see a blunted activation of more pathological gene sets. So a lot of these genes are activated in the failing heart, and these responses were blunted with our knockouts. We also saw a reduction in lung edema, which is another indicator of heart failure, fluid accumulation in the lungs, which was reduced in our knockouts as well. Now, we also did another study, which I think is even more impactful, which was genetically intervening and deleting glutamine synthetase after the establishment of heart failure to see if this would either reverse or slow disease progression. Because again, this would be a much more clinically relevant and interventional type of therapy to treat heart failure. And so here we induced pressure overload for 8 weeks, followed by the provision of tamoxifen to effectively delete the gene, and then followed them out for another 12 weeks. Again, we saw there was no difference between the stress that these two groups were under based on the peak pressure gradients across the aorta. But as you can see at the time of tamoxifen administration, prior to that, both these groups tracked very nicely, starting to develop heart failure. But as soon as we provide tamoxifen and delete this gene, the GS knockouts do significantly better than the controls. And so we're kind of working with two hypotheses right now of why we think this glutamine synthetase knockout is beneficial. One of them being this energetic effect that is occurring within the cardiomyocytes. By deleting this, we have a therapeutic benefit by increasing energy metabolism and kind of plugging this potential leak of carbons out of the Krebs cycle. Um, and then the second hypothesis potentially is that this glutamine could actually act in a paracrine signaling fashion to activate those myofibroblasts. So starting now to kind of link part one with part two of the talk, we think that this could be a really novel and interesting phenomenon whereby the cardiomyocytes are actually supplying the glutamine fuel to the fibroblasts to promote fibrosis. And we do have some data that would support this. We see that when we look at the activation of the fibrotic gene panel within our mice subjected to TAC, we see in the blue bars, we see an activation and increase in a lot of these fibrotic genes. However, when we delete GS in the green, you can see that there's a significant reduction indicating less fibrosis in these hearts. When we do our Picross series red staining again, you can see that in the Cree controls, very little fibrosis in the sham, much more fibrosis, red staining in our TAC animals, which is completely abolished and prevented in our knockouts. And so this really just asks the question of, could cardiomyocyte-derived glutamine signal and initiate a fibroblast to myofibroblast differentiation? And that's something we're currently trying to work at in the context of some complex and pretty cool in vivo studies. So as Dr. Cook mentioned, I've only been here for about a year, but this is kind of the main focus of my lab right now. Several projects focused on cardiac energy metabolism, the activation of fibroblasts and processes that we can mediate and reverse, as well as metabolic and transcriptional regulation of heart failure, and then some other stuff that I think is really cool, but more to come on different organelles and compartments of the cell talking to each other in the context of heart failure. So with that, thank you all for coming and to those online. And I would like to thank all my collaborators and people that helped with these studies, and currently the two very good individuals in my lab as I begin my career here at Louisville. So thank you.
Really fantastic, Dr. Gibb. Can you hear me? I just have one major question for you. So it's actually good to hear the name Howard Techmeyer. He was my biochemistry professor at med school. Wonderful guy. The question is another University of Chicago issue, which is we came up with myocardial perfusion imaging with nitrogen-13 ammonia. It was easy to synthesize, and it seemed to track blood flow pretty well. It rests in stress. 1977, advanced a few years, and yeah, it got overdone by thallium and rubidium and all that stuff. But we're about to embark on cardiac PET. It's been part of our capital equipment purchase. Just has to wait for a big building to be finished and that sort of thing, but it will come to UofL. And nitrogen-13 ammonia is actually one of the tracers that will be competing for our business. Now, the funny thing about it, two things. One is that it gives you an infralateral perfusion defect that nobody understands. But the other thing about it is that it isn't exactly just perfusion. It is actually glutamine metabolism. And so I'm wondering if we have a PET program with nitrogen-13 ammonia, how can that be used to further your work? The one thing, when we do perfusion imaging as a stress test, we're doing the stress image and then a resting image, and we could do so much more. We could actually follow the characteristics of the decay or the metabolism or the washout. There's so much more that we could do by continuous imaging over a period of time. So I just had a thought. I had to try to pick your brain about how our PET program with nitrogen-13 ammonia could actually help in this investigation that you're doing.
Yeah, I think that sounds really interesting. Glutamine is actually a potent vasodilator. And so I think that there's definitely a potential there for kind of what you're talking about, right, with PET imaging and trying to understand how that's occurring. I actually just had a really interesting result yesterday where I exercise, the knockouts, in comparison to the controls. And the hypothesis was that they were going to run a lot farther because they were more energetically capable and had this higher energy state. And they actually ran not as far, significantly not as far. And so, potentially, maybe that decouples it a little bit from energetics, but, maybe there's some other paracrine signaling factor here that I'm missing, that's in addition to the fibroblast compartment. But something that maybe, deleting this gene or glutamine derived from the cardiomyocytes is really important in hyperemia. So I think that's a really interesting thing that, could maybe tie into some of the stuff you're talking about.
Oh, it'll certainly be interesting. Any other questions? I can't see if the people who are there or anyone who's online, welcome you to unmute.
There's a question in the chat area, Dr. Gibb, from Dr. Dryden and GI.
Okay.
And he says, great talk. Do you think that pharmacological inhibition of GDH will be as effective as genetic deletion?
Yeah, I would say probably, I guess it would be possibly similar. But, because the idea would be like, if you're still deleting glutamine, could you be deleting or preventing glutamate utilization, I guess could be one of the questions, right? But it seems like most of the glutamate is coming from the glutamine. So I think it would be as effective. It could just be, another further downstream effect. Yep.
So one thing that stuck out to me, I don't quite piece together is, so piecing these two things together, right? The paracrine kind of theory. So if I take the fibrotic heart and I intervene with glutamine, however that was, I know I'm not an expert, I can't really remember, but you intervene with glutamine is what I'm putting in my head as, and your diastolic function gets better, correct? Your fibrosis goes away, your diastolic function gets better. But then you flip over and you work on the glutamine synthetase and make the energy production better. It seems that it's only a systolic issue and the diastolic did not change. At least the LVID didn't change, right? And so why would it be that the paracrine thing makes sense if you think that the glutamine is going over and intervening on the myofibroblast, but the diastolic parameters are not improving at all?
So I think the idea is that, the cardiomyocytes don't seem to use glutamine. Okay, they seem to make it. And then the fibroblasts really like it and they use it a ton, right? So by intervening by, say, using that pharmacological inhibitor, right, to prevent utilization, it kind of works really well, right? Because it's going to target that fibroblast population that really, really likes to use it, but it is unlikely to really affect the cardiomyocytes because they don't seem to use it, right? Now, the other side of the cardiomyocytes making it, right? So making it, then it's kicking it out, and the fibroblasts are like, oh, this is great, right? It's right here. I'm sitting right next to you. I can use it. Or there's, different connection junctions that link these cells together, right? So they can kind of just feed it to them. Now, deleting that gene would prevent fibrosis, potentially, right? Prevents fibrosis by preventing that paracrine factor, but it increases energetics.
But it didn't change the diastolic function on the echo, right? Because the LVID and things like that were the same. You said specifically that it was mainly a systolic derived benefit from the blockade of the GS, right?
Yes, in the cardiomyocytes.
So then why would it not have a diastolic benefit as well? I don't understand if the fibroblast is involved.
Yeah, the diastolic function is improved. It just didn't, reach, but you can see they separate, right? Yeah. So it could just be that, or it could just be like our measures for diastolic function weren't as sensitive.
The second part of my question is, what do you think about statistical benefit versus clinical benefit? So I noticed like in your ejection fraction change, things like that, at least in the first section, you're talking about things like an EF change from 50 to 55, right? So what is the expected clinical improvement from the changes that you're seeing in magnitude and what you're doing? Do you have a comment?
Yeah, that's a really good question, right? I think that's something that all of us stress and think about, right? Is that we can do a lot of things that make a actual benefit, but is it clinically meaningful, right? And the two most important things I would think in the clinic, right, are like quality of life, right, and mortality, right? Because if you can't improve those two, then it's kind of, what are we doing? And so we'd have to really kind of test that out and see if it does make a big difference, right? And it could make a huge difference in certain etiologies of heart failure, right? And say an MI where we have this kind of really big scar, right? That's important in maintaining that structural integrity. intervening and inhibiting fibroblast formation could be really, really bad, right? Really, really detrimental. And so I think we'd have to really kind of tease that out. And it's kind of also, what if we intervened earlier? Would we have a better, improvement in function? That's something we'd have to look at.
Yeah, I think that's another hard thing. I mean, most of these people come to the hospital or get diagnosed.
They're already there,
because
right?
they're there,
Yeah.
right? You're not going to intervene halfway down the road.
Yeah.
The last question I had was about unintended consequence. Anytime you start intervening on a mechanism that is a fibrotic mechanism, things like that, there are possible unintended consequences. Are there possible unintended consequences of intervening on a myofibroblast? For example, why does the myofibroblast exist? Is it just a pathologic thing, or are there benefits?
Yeah, so there's a lot of research in myofibroblast on those types of questions, right? So some of them become potentially these matrix fibrocytes, which become a little more contractile and actually functional. So I think, they're definitely necessary if we talk about it in the context of an MI, right? Really important that they're there, because otherwise you're just going to rupture and bleed out, right? So in that context, really, really important, right? But in the context of, say, pressure overload or HFpEF or things like that, where it's more progressive, I don't know exactly what the role is, and if we do get rid of them, is it good? There's been some studies where they've deleted them like since birth, and the heart does fine.
So in one of the slides, you showed the effect of TGF-beta and angiotensin 2. And I forgot, was it fibroblast or myocyte?
Fibroblast, yeah.
Fibroblast. And can we go back to that slide?
Maybe.
Might have did the screen share. Oh, there you go.
Let's see. Oh, don't crash.
Well,
Yes, here you're seeing 10% improvement in ejection fraction with the deletion of the fibroblasts. So that, to me, that seems a pretty decent improvement. Go back. So what was the question?
The question is that the angiotensin 2, you showed TGF-beta, they were in fibroblasts, right?
Yeah.
And they were increasing the fibrosis or production of glutamine.
So stimulating fibroblasts with those pro-fibrotic agonists increases glutamine utilization. Yeah.
I see.
And so the question was, if we can block that metabolic pathway, can we then prevent the fibroblasts from activating and becoming myofibroblasts?
Did you try to block angiotensin 2 function by blocking the receptor?
That's all been worked out very extensively, yeah. That the TGF-beta receptor and the angiotensin receptor, that these are known fibrotic activation pathways. Yeah. Yep.
That's what I saw in my research, that angiotensin 2 induce fibrosis left and right ventricle.
Yeah, and that's a model of like inducing fibrosis in mice is using like pumps that you can implant with angiotensin 2 and phenylephrine and these other types of things that induce a fibrotic response. Yep.
So is the plan to have the glutamine blockade or whatever? There's a pill, I think, or some treatment that exists. Is that like a daily thing, or?
I'm not exactly sure how they do it in the cancer clinical field right now, but it is in phase 2 for tumor suppression, but nothing's been kind of worked up in other.
Because I guess the natural question is that this is a ongoing daily treatment, or infusions, or this or that, and we know patient adherence and all those things are issues.
Yeah.
Why not just kill the fibroblasts? Why are we trying to block it every day?
Yeah, so there's been some studies that use like CAR T therapy type stuff within fibroblasts.
It's from Penn, right?
Huh?
Yeah, I think the first one was from Penn.
From Penn, yeah. Paul Epstein. Yeah. So he's done some of that work, and he's presented it. I think it's published a little bit, and it's already in scientific companies and stuff like that they're trying to work. But they do still come back because there's still populations, right? So it does become a maintenance thing.
Yeah.
But with the CAR T, it might be go to the clinic for an injection every few months or something, right? Which would definitely improve. But even just a pill, while it's not 100% compliance, it's still better than.
Or maybe it's CAR T and then take this pill.
Yeah. Yeah, lots of possibilities for sure. So.
Yeah, the cancer cells are smart. If you cut down the glucose, they use glutamine as a source. They never die.
All right. Well, thank you guys.
Thank you.